So now that we have explored an overview of the role of nanopore sequencing in microbiology applications, let's take a closer look at the technology itself. So what you can see here on the left-hand side of the slide, right? These are the surface of the floor cell. So it depends on the floor cell they are using. There are hundreds to thousands of these tiny holes, which is a nanopore embed uh in a what we call a electro-resistant membrane. And each of these nanopore will correspond to its own electrode connected to a channel and a sensor chip, which measure the electric current that flow through the nanopore. And during the sequencing, the electrical current will be applied. And when a molecule passes through a nanopore, the current will then be disrupted to produce a characteristic squiggle. From what you can see from this image here, this is what we call a squiggle. And then the squiggle will then be decoded using a base calling algorithms to determine the DNA or RNA sequence in real time. Or to make it easier to remember, you can think of the current as a water that flow through the pipes. So when an object enters the pipe, the flow of the water is disrupted, just as how the DNA or RNA disrupt the current as it's passed through the nanopore. So when a strand of the DNA or RNA that make up of a sequence of a different combinations of the nucleotide bases A, T, G, C. So when each base that pass through the nanopore um, cause the interruption to the signal, this characteristic interruption will then be converted to the base call. So this way of sequencing is what makes nanopore sequencing very unique because it allows a direct real-time analysis of DNA or RNA and including the modification informations because when the base includes modification informations, the patterns will be different and that is how our base calling algorithm are able to differentiate these signals. And what are the unique benefits of nanopore technology offers? So first of all, it allows the direct sequencing of DNA and RNA. So the benefits of doing the direct or native sequencing of DNA and RNA is that it can retain the epigenetic informations. And in fact, nanopore sequencing platform is the only sequencing platform in the market that allows you to do direct RNA sequencing without the needs to convert your RNA to cDNA with reverse transcriptions. So there are a few advantages of doing a direct RNA sequencing. So first of all, you get to study the epitranscriptomes, um, which is not possible with the cDNA approach. And also with the direct RNA, you can avoid the bias and errors introduced during the reverse transcription process, especially those with a complex secondary structures which may not be efficiently reverse transcribed. And in some cases, right, the reverse transcription failed to cover the full length of the RNA molecules, which result in incomplete transcript. So, in addition to that, you also get the benefits for preservations of the strand information. And because of us being able to do a direct sequencing of DNA and RNA without the involvement of PCR, there's no amplification bias. It also means that the entire library preparation workflow is much more simpler and much more streamlined. And we are also read length agnostic, although we are often being perceived as a long read sequencing company. But the fact is that we also have the short fragment mode that is integrated into the operating software that allows the sequencing of fragment as short as 20 bases on the Nanopore platforms. So in other words, we can sequence as short as 20 bases to as long as you need for for your for current records, the longest the single strand of DNA that ever to sequence on the Nanopore platform is 4 MB. So it's really depends on what is the length of the library that you fit into the device instead of having to limit, what kind of length that you should put into the sequencer. And also, we are the only sequencing company that allows you to do real-time analysis. So you can set like every half an hour or every 1 hour with the data generated, you want to do analysis and see whether or not the data generated is already sufficient to answer the questions. And if yes, you can choose to stop the sequencing run, wash the floor cell, and then keep it in the fridge so that it can be reused for your next experiments. So this is really a unique features that only offered by Nanopore technology. And also we give you the options for both devices and floor cell from a smaller format to a large format. So it means that you can run your experiment very uh, cost effectively according to your sequencing needs. So in this slide, this is a summary of the relationship between our floor cell and device. Um, Here on the left-hand side, the mean ion and grid ion devices runs two types of floor cell, a Flonger and mean ion floor cells. So the mid-ion floor cell have a theoretical maximum output of 48 gigabytes, and the scientific community have been always constantly trying to, work towards this theoretical maximum. However, for now, you can typically the throughput that we observe is usually in 10 to 20 gigabytes, depending on a number of different factors like the sample quality, the quality, as well as the N50 etc. And then on the right-hand side, these are the Promethean series of device with theoretical maximum of over 270 gigabytes. And currently, you can expect between 90 to 150 gigabytes with the human whole genome sequencing. And we are constantly improving our chemistry, our floor cell to bring you to this theoretical maximum. And another thing to highlight is that it really depends on your throughput. You can start with a very small device running one floor cell at a time. And if you have higher throughput, you can have a grid ion device that allow you to run 5 floor cell at a time. And for Promethean, we have Promethean 2, which allow you to run 2 floor cell at a time. Um, Or if you require a higher throughput device, we have P24 or P48 that allows you to run 24 to 48 floor cell max at a time. However, one thing to highlight is that all of our sequencing device allow you to run each floor cell independently. It means that you can, for example, for P48, you can sequence anywhere from one floor cell to 48 floor cell without requiring any batchings. So in terms of the cost effectiveness or cost efficiency, you will not be getting penalized for not having enough samples to batch in a certain scenario. And with Nematode sequencing, you can perform the real-time targeted sequencing through what we call adaptive samplings, or use this technique to reject targets that are not of interest. Like, for example, if you want to exclude the host genomic DNA in pathogen surveillance. So, what is adaptive sampling? So, this is actually a method which allows you to enrich or deplete a region of interest using software. So, what you really need is a bag file which contain the region of interest and as well as a FASTA reference file. So once the sequencing started, right, due to the real-time nature of the nanopore sequencing, our software, our operating software Mino, will be identifying and then to know that whether the strands that's being sequenced is within region of interest. And in 1 second for Nanopore platform, approximately 400 to 800 base pair of bases will be sequenced, depending on which device. And with real-time base calling and alignment, if the reads does not map to the region of interest you define, then the software will eject the DNA strains from the nanopore. So then now the nanopore will then be available for the next strain to come in. And the off-target strain are going to continually get rejected until your region of interest is being detected and the sequencing is allowed to proceed.
So just want to have a quick question. So this method, no adaptive sampling can you for any of your machine or limited to some?
All of the machine. You can do it on MinION. You can also do it on the Promethean floor cell.
Okay, okay,
Mm,
thank you.
yeah. So with the adaptive samplings, you can potentially design an assay, one single assay that allows you to do SNP detection, allow you to do indels, allow you to do structural variants as well as methylation. Because with adaptive samplings, right, in terms of wet lab, you do not need to design any primers. There's no involvement of the PCR. There's no involvement of target capture with hybridization. All you really do is really just supply a back file and a FASTA file and telling the software which is your region of interest. So in terms of the workflow wise, it's exactly like whole genome sequencing, which is less than 60 minutes of the library preparation time, then you can do adaptive sampling. And also remember we mentioned about in the adaptive sampling, there's a lot of rejection that happens, right? And all these rejections that happens will be your off-target reads. However, the off-target reads can be useful as well because it can be then used for low-pass genome-wide applications. And one of the use case of these adaptive samplings in, for example, in human application is that you can design a targeted sequencing panel that have, for instance, pharmacogenomic genes or hereditary cancer genes. And so you will get a good coverage step depending on how many coverage step that you set, 30x, 50x. So you get that. At the same time, you also get this off-target reads for, low-pass genome-wide analysis. Then you can actually do genome-wide imputation for PRS application. So this is really a very powerful tool that a lot of customers are very interested to optimize it to develop assay.